just testing.
It's 11 o'clock, so we're going to go ahead and get started. Hi, everyone. It's 11 o'clock, so we're going to go ahead and get started. The Center for Integrative Environmental Health Sciences and the Department of Pharmacology and Toxicology welcomes you to our Environmental Health Science Series seminar. It's my pleasure today to introduce Dr. Barbara Clark. Dr. Clark got her bachelor's degree in zoology from Miami University and her Ph.D. in biochemistry from the University of Texas Southwestern Medical Center. From there, she went on to get a postdoc in endocrinology at Texas Tech University Health Science Center. And following that, she joined the Department of Biochemistry and Molecular Biology here at the University of Louisville. Following that, she's held several positions within the university, including acting associate dean of the graduate school and her current role as the associate dean for graduate postdoctoral studies and as a program director for IPIBS. In addition to her administrative roles, she has been an active educator. She has worked as both a course director and a lecturer for multiple courses within the graduate school. And she has published more than 50 first author or co-authored papers and has held positions in various national grant review and editorial review boards. Today, Dr. Clark will be giving a talk on the cholesterol transporter Stard5 and its potential role in a new mouse model to study fatty liver disease. And with that, I will hand it over to Dr. Clark.
Well, thank you so much for that nice introduction. And it's my pleasure to be here. I'm a little short, so if you can only see the tops of my eyes. I am really excited to present some of this new work, and I've always had a problem with long titles, but really what I want to pose to this group and to get feedback from whether the Stard5 knockout mice that we have developed could be a potential new model for study of fatty liver disease. And the work I'm presenting has really been the work that has been supported by the CIEHS, and that is first through obtaining a voucher award, and that was followed by a pilot study with my co-I, Carrie Kling, where we're studying Stard5 and lipid dysregulation with endoplasmic reticulum stress. So what I would like to do first is introduce this group to the Stard protein families. And so we'll give a lot of background on that. And then we'll be focusing on endoplasmic reticulum stress and really how that is linked to non-alcoholic fatty liver disease and where I want to go in the future with this work. So Stard5 is a member of the Start domain superfamily. And this is named for the founding member, Stard. Stard stands for steroidogenic acute regulatory protein. And when we first identified this, it'll be 30 years next year, which is amazing to think about. When we put it into a database, and when I say a database, there really weren't any databases around. This was a novel protein, but after several years looking at the sequence of the Stard protein, they found that there was a 200 amino acid stretch that had sequence similarity, but more importantly, what was shared from multiple organisms that was a Start domain that shows this alpha helix grip fold. And what is common to all these proteins across the phyla is that this fold allows for this, what is depicted here, a hydrophobic groove. This is going to be the binding site for lipids, whether it's sterols or phospholipids. And also what is important for this family of proteins, at least we know for a few of those, this is a amino terminus and a C terminus. This alpha helix, as you can see, is depicted here in the structure, aligns on top of this hydrophobic groove. And so it's a movement of the C-terminal alpha helix that is important for lipid binding and lipid absorption and desorption, I would say. So this family of proteins is known as START for the star-related lipid transfer domain proteins. And what I will show you here is that the mammalian START protein family. What is common, again, to all the protein family is the START domain, which is depicted here in this cartoon as a lipid binding domain. And the different subfamilies, there are 15 members within the mammalian start domain family. The different subfamilies are characterized by their ligand binding. So in the case of the founding member Stard1, that binds cholesterol, as well as this Stard3, which is also MNL64. And I'm saying those names because in the literature that has historically been used, and what is unique about this subfamily is that there is a signal sequence. In the case of Stard1, this is a mitochondrial targeting sequence. In the case of Stard3, this targets it to the endosomes. So these proteins are directed within the cell. They function at the site of where they're directed. The next subfamily, which includes Stard5, is the other two members are Stard4 and Stard6, are composed solely of the start domain. So there's no targeting of these proteins that we know of to any cellular organelle. And these start domains do bind multiple sterols, including cholesterol. Then we have the phospholipid binding proteins. As you can see down here, this is the phosphatidylcholine is a major ligand that has been identified to bind to this subfamily of proteins. And then there is this ceramide binding protein, or Stard11. And the family, including Stard12, 13, and 8, these were identified as tumor suppressor genes that are deleted in liver cancer. And what is unique about this family, or I should say in these types of families, is the star domain is part of one domain of a multi-domain protein family, where you can see they have extra activities, such as the ROGAP activities in the start domain family. At present, it's not known whether ligand binding to the start domain within the Stard12, 13 subfamilies is important for its function, although the ROGAP function is important for tumor suppressor activity. Then we have, again, another multi-domain subfamily, Stard14 and 15. Here they have these hot dog motifs, and that the hot dog motif is what is described for acyl-CoA thioesterase activity. So they fold into a certain fold promotes the thioesterase activity of this subfamily. And then we have this lone partner, Stard9, that has not been well characterized. So again, commonality of this is the start domain of binding lipids. And what we do know is that Stard5 binds cholesterol, and we will be focusing on Stard5 for today. So what are the model? I just want to emphasize, I guess, also that, as I indicated, that we have more information on some of the first protein family, that is the Stard1 and Stard3, because as the signaling sequence that targets Stard1 to the mitochondria, it will be imported into the mitochondrial process as a typical cytoplasmic translated protein that enters the mitochondrial matrix. However, even though it's been 30 years, the function of getting cholesterol transport across the mitochondrial membranes, and I should say the import of cholesterol into the mitochondria is critical for production of steroid hormone biosynthesis. The first enzymatic reaction for estrogen, testosterone, mineralocorticoids, and glucocorticoids requires this transfer of cholesterol into the mitochondria. And it is essential that STAR be synthesized to mediate that transport. Although it is targeted to the mitochondrial membrane, it's not its import that's important for function, but it is required that you have an interaction of the start domain with other mitochondrial membrane proteins, indicated here is VDAC2 or PCP, and there is a complex that is formed between the outer and the inner membrane that allows for cholesterol to transport through the membrane. I say this because here if we delete 10 amino acids from the C terminus of Stard1, we lose all ability to mediate cholesterol transport into the mitochondria. So that gives us evidence that the C-terminal helix is very important in maintaining the binding and the absorption and deabsorption of cholesterol at a membrane. Stard4, as it says, targeted to the late endosome, so there's transmembrane regions, and it has other motifs such as the 4-amino acid fat motif, which is important for interacting with the ER resident membrane protein, the VAMP associated protein, the VAT proteins. So what this does is it tethers really the late endosome to the ER membrane, and cholesterol transport between these two membranes is dynamic. And cartoon here is the start domain, so you can see that you have cholesterol can move between these two membranes by binding to the start domain of Stard4. Similarly, for ceramide transport, it's a similar type of mechanism, except in this case, you have a plextrin homology domain that will interact with phospholipids and specific membranes, in this case, the Golgi. You actually have that fat motif that will then tether this start protein to the ER. So here again, we have set up the situation where we have interaction between the Golgi and ER membrane by tethering this start domain, and that leaves this start protein, leaving the start domain free to move ceramide from the ER to the Golgi complex for single lipid synthesis. So we can understand the movement of cholesterol and lipids between those membranes based on the other motifs within the start domains. But the challenge that we have with these soluble cholesterol transporters, which are the Stard4 and Stard5 subfamily, is that they are composed completely of the start domain. So there's no information in that primary sequence or secondary sequence to indicate that these proteins should associate with any particular membrane. So what is shown here is that C-terminal alpha helix of the model that continues today is that there is a donor membrane, and if you overexpress either of these proteins with vesicles, you can have cholesterol moved from a donor membrane to an acceptor membrane. So the idea there is you have absorption from one membrane, it's protected in the cytoplasmic space, sequestered in the interior of the transporter, and when it reaches the acceptor membrane, you have deabsorption into that membrane. The question is, what is the donor and what is the acceptor membrane for these soluble start proteins? And I will show you data on how we think this moves. This is showing within the cell and this is only focusing on the start proteins, because there are other proteins within the cell that also play a role in non-vesicular sterile trafficking. And I emphasize this as non-vesicular because we're well aware there's vesicular types of transport through Koch vesicles that can move sterols between membranes. But when we think of soluble transporters, and these are in the start domains, I've indicated here the Stard4, where we maintain cholesterol between the ER and the late endosome. We're moving ceramide from the ER to the Golgi complex. Data has shown that Stard4 is highly associated with the endosome recycling complex. So this is really a reservoir of cholesterol within the cell. You can have uptake of cholesterol from the plasma membranes or lipoproteins coming into the endosome recycling complex that can be reshuttled back to the plasma membrane. You have delivery of cholesterol from the early lysosome or late endosome to the endosome recycling complex. We also look at how cholesterol can be moved directly from the plasma membrane to the ER. And here at the ER, one of the roles of Stard4 has been shown to activate the cholesterol acyl transferase, or ACAT enzyme, that converts cholesterol into cholesterol esters, and so you can form the lipid droplets. We also know that there's movement between the ER and plasma membrane that is mediated by Stard5, and some of the most recent work has been showing that Stard5, when it moves cholesterol to the plasma membrane, it may enhance cholesterol efflux through ApoA1 mediated efflux to forming HDL. Now, what will provide some of the data that supported how we are thinking about Stard5 and its function of moving cholesterol from the ER membrane to the plasma membrane. But I will say there is very limited data on the mechanism or the trafficking of the soluble Stard5 and Stard4 proteins. And most of that has all been in vitro work. And why is this important? Well, we have to maintain specific distribution of cholesterol between the membranes within the cell. And shown here is the estimate of the total cholesterol that is with the plasma membrane. We say majority of the cholesterol is localized in the plasma membrane. We have very small amounts in the endoplasmic reticulum, and the Golgi is somewhere in between. And this reservoir recycling complex has about 35% of the cholesterol. So it's important we maintain this distribution, and when we focus on the ER to the plasma membrane, if we have an overabundance of cholesterol in the ER membrane, we will look at that in a minute, how that can disrupt signaling. So the question that I have is does Stard5 play an important role in maintaining this distribution of cholesterol between the ER and the plasma membrane? And if it's dysregulated, does that contribute to some disease states? So that's it. So why Stard5? Why am I interested in non-alcoholic fatty liver disease? Well, the first question my career has been built on studying the Start protein family of members. And Stard5 struck my interest because earlier work that we did looking at the kidney, we were able to show that Stard5 is overexpressed in diabetic kidney. And there's been data in the literature to show that Stard5 expression is induced by ER stress. And I've always made the argument that Stard5 moves cholesterol to the plasma membrane for efflux. And if we think about this, ER stress and the cholesterol accumulation in the liver are hallmarks of non-alcoholic fatty liver disease. And why NAFLD? Well, I just look at this group and the expertise at this university, and this is a great resource and an important disease to begin to get some ideas on novel targets that we may have for treatments. I do want to point out that I did read the announcement that Dr. Kling sent forward to me that the new nomenclature is steatotic liver disease, or SLD, or metabolic dysfunctional associated steatotic liver disease, which I think will be MASHLD, I'm not sure, and metabolic dysfunction associated steatohepatitis, or MASH. I didn't have time to change all my acronyms, so I'm going to be using NAFLD and MASH for this talk. So the question really here is, does Stard5 help to reduce cholesterol accumulation under conditions of ER stress? And ultimately, to apply the knowledge to determine whether Stard5 can reduce cholesterol accumulations under a diet, environment, toxin exposure. So why ER stress? Well, I start with ER stress and looking at the impact on lipid accumulation. That's because there's a historical link between ER stress and non-alcoholic fatty liver disease progression. And which is just shown here is that we will be looking at the ER stress pathways again in a couple slides, but really the major mediators, here's PERK, IRE1 alpha, and ATF6 alpha. Activation of these ER membrane proteins due to exogenous ER stress can result in transcriptional changes. So in broad strokes overviews, what activation of these signaling pathways downstream of these ER activators, then you can have results in lipid accumulation. That's what we see with the steatosis. And looking at specific target genes, you can have an increase in lipogenesis, increase in uptake of fatty acids, increase in VLDL production, and I've just highlighted here also an increase in free fatty acids. Now you can have adaptive ER stress and you can resolve the steatosis, but if you have continued ER stress, as you can see cartooned here, continuing increase in ER stress, this can lead to macrophage recruitment, inflammation, and increase of the signaling pathways within the hepatic cell types that will increase flow-inflammatory responses, oxidative stress, and that can lead to cell death. And then in a very small subset of patients, it's been shown that this can further progress to hepatocellular carcinoma. But we are going to be focusing really here on this first step of development of steatosis and interested in what factors may help trigger or advance that disease onto NASH. So Stard5. Stard5 is an ER stress responsive gene. And this is when the first discovery of this subfamily of the Start proteins. So one of the studies that were done is they isolated mouse macrophages and they loaded them with cholesterol ester or free cholesterol to induce ER stress. And what is shown on this is a PCR looking at XBP1, which is a downstream signaler of activated ER stress. And when differential splicing will occur on ER stress, leading to a higher, larger transcript, which is shown here. So we can see in the free cholesterol-loaded macrophages that we have induced ER stress, but not in the cholesterol ester-loaded macrophages. Dept is also known as GRIP78, which is the ER stress response protein, and we can see under conditions of free cholesterol loading, we have an increase in BIP expression, which is one of the early responses, as well as an increase in Stard5 expression. But we don't see an increase in Stard4, rather we see that it is suppressed in the presence of this ER stress. Focusing on Stard5 with cholesterol ester loaded, we don't see any effect. And then using NIH3T3 cells, again using a chemical inducer of ER stress, tunicamycin, and we'll see this is what I've used. Tunicamycin prevents the N-glycosylation within the ER. That results in the buildup of proteins in the ER that triggers the ER stress response. And again, here we can see that in these cell types, we get an increase in ER stress as indicated by the induction of GRIP78 as well as Stard5. And similarly, using multiple mechanisms to induce ER stress, as I said, tunicamycin, thepsogardin, blocks calcium efflux from the ER, so that will induce ER stress, and Berfeldin A blocks trafficking from the ER to the Golgi. So all these mechanisms will induce an ER stress, as we see here, through the induction of the GRIP78 protein, gene expression, and they all induce Stard5. So Stard5 is mirroring the classical marker of ER stress, I would say, in terms of ability to be induced by ER stress. Now, from our work in the kidney cells using the human proximal tubule kidney cell line, HK2 cells, we also saw that with tunicamycin and thepsogardin that we were able to see an increase in the ER stress as well as an increase in Stard5 protein. So it's very clear that in cell lines and cell models treated with these ER stress inducers that we have an increase in Stard5 mRNA expression. And then looking at Stard5 cellular redistribution with ER stress, that was a question we asked because we are looking at is ER stress, if the proposal that Stard5 is moving cholesterol from the ER to the plasma membrane under ER stress, can we see redistribution of the protein? Again, from our work, we're using the HK2 cell lines treated with tunicamycin for 24 hours. But we observed the green fluorescence using immunohistochemistry or fluorescent chemistry to mark Stard5 with the green here. We can see that in the control cells, we have pretty broad distribution of Stard5. But with tunicamycin treatment, we can see that we have distribution or movement, I would say, to the plasma membrane of these cells, as well as some punctate staining around the perinuclear regions. So we also want to know, well, what organelle or membranes might Stard5 be associated with. So we use markers for the endosome, early endosome compartment, and GRIP78 for the ER component. And here you're seeing Stard5 in red. And when we looked at the merge image in the control cells, we don't really see any overlap. But again, here we can, with the early endosome marker, but with tunicamycin treatment, we do see with this yellow staining that we have redistribution and overlay with the early endosome. Similarly, when we look at the co-localization with GRIP78, we can see that with tunicamycin-induced ER stress, that we have more prominent co-localization, and we can see this tracking to the plasma membrane as well as dense around the perinuclear regions. So these data convinced us that with an ER stress, Stard5 is moving within these cells. So for the group out of Virginia Commonwealth University that has also been working on Stard5, they have used primary rat hepatocytes. And in this case, they overexpress Stard5 in those hepatocytes and use polypin to stain cholesterol. And if you look at these images, you can see this is, and Stard1 is the negative control in this case. You can see brighter images, what they quantified down here, showing that when you have overexpression of Stard5, you're accumulating cholesterol within these hepatocytes. And then they took those cells and did membrane isolation, subcellular distribution, to ask whether there's a change and where Stard5, or I should say where free cholesterol was recovered within the membranes. And it's shown here in the control cells. This is shown the pellet and then separating into soluble or the microsomal fractions. So this is a high-speed centrifugation, so you bring down the microsomes and probably plasma membranes in this case. That when you have Stard5 overexpressed, as shown in the black bars, that you have this equal distribution. Now you're seeing more prominent distribution of cholesterol within the microsomal membrane fractions, suggesting that Stard5 is promoting this redistribution of cholesterol within the cell. And most recently, from this group, what they have generated a Stard5 knockout mouse. This came out when we were developing our Stard5 knockout mouse at the same time. They haven't fully characterized this mouse model, but what they were very interested in, it was asking the question, you know, what does the absence of Stard5 do to movement of cholesterol within the cell? So first, if we look at this, we look at the wild type in the knockout mouse model, and these are primary mouse hepatocytes isolated from either the wild type or Stard5 knockout, treated with thapsogardin to induce ER stress. They use CHOP to show that the ER stress is induced in both the genotypes, the wild type in the knockout model, and that with their thapsogardin-induced cell line, they see an increase in Stard5 protein expression. They then ask the question, does ER stress or the presence of Stard5 impact plasma membrane cholesterol levels? And to measure that, they use this protein from, I believe, bacteria that basically can bind to cholesterol in the outer leaflet of the plasma membrane, and this is a fluorescent detection. So this is looking at this binding, increase in binding of this, I would say probe, would be an indicator of increase of plasma membrane cholesterol. And what you can see if we look at the wild type animals, that you can see with ER stress induction, they saw an increase in accessible cholesterol at the plasma membrane. But when they looked at the knockout mice, there was less cholesterol at the plasma membrane, and that in the presence of ER stress induction, there was no movement or no greater increase in plasma membrane cholesterol. So based on this, the model has been supported that Stard5 is promoting the movement of cholesterol to the plasma membrane. We can't say it's from the ER membrane in this case, but we can say that there's an accumulation of cholesterol in the plasma membrane in the presence of Stard5. So where is Stard5 expressed? Well, based on the Human Protein Atlas, it's everywhere. What we know from the literature is it's present in the liver, and this is where it's been most studied. It's also in macrophages, the Kupffer cells of the liver, hepatocytes, and I believe also in the stellate cells of the liver. Macrophages, studies have been done using the THP1 cells, and we have shown that the expression in the kidney, as shown over here, is restricted to the proximal tubule cells. This is a IHC for Stard5 in mouse kidney. You can see there's absent in the glomeruli, but highly prevalent in proximal tubule cells. And I'd like to point out that we see it is very concentrated at the apical membrane. So there's it. Also, looking at rat small intestine, we did IHC also, and again, we see that Stard5 is most prominently localized to the microvilli and at the apical membrane. So this is indicating that it might be in the right spot for cholesterol efflux. So the reciprocal relationship between lipotoxicity and ER stress. So we know, so this shows Dept. So this is Dept or GRIP78. This protein is induced upon ER stress, whether it could be genetics, environmental, or chemical. Dept is the one that promotes the activation of the ER resident proteins that activates downstream signal transduction pathways. We're not going to go through all these pathways and specifically, but just to give you a sense that each arm of the ER stress response can activate different pathways. But they can work in concert to provide increase in lipogenesis, or they can also provide a decrease in lipogenesis and gluconeogenesis. So many times it depends on the inducer, the context, the cell type, and the combination of activation of these different arms of the pathway, what your ultimate outcome will be for the ER stress response. So what we do know, in the context of NAFLD, is that if you have a high-fat diet and you have increase in lipogenesis ongoing, lipogenesis can be a trigger for ER stress. Conversely, if you have a chemical insult that induces ER stress, you can have an increase in lipids in the liver. The increase in lipids then will promote further ER stress. So this is the lipotoxicity and ER stress is this conundrum. You know, it's a vicious cycle, and so trying to block that cycle could be an important pathway for looking at these disease states such as NAFLD. So now to the work that we have done supported by the CIEHS. Again, the question is, does Stard5 help to reduce cholesterol accumulations under ER stress? We've generated Stard5 knockout mouse line using CRISPR-Cas9, validated there's no RNA or protein detected, and used those founders and did 7 generations back cross onto C57 black 6 mice. So using 2 to 3 month old male mice with littermate controls of the Stard5 knockout mice, divided into 2 groups, either DMSO control or tunicamycin. There was a single injection of tunicamycin given IP to the mice, and then 4 days later, we collected the liver tissue, and they were processed for histology, RNA to do RNA-seq analysis, and for protein for Western blot and lipid measurements. This is just to confirm, if you're looking at our Stard5 knockout mouse model, you can see with the wild type, which is in the black, and the knockout or in the red. If we have the DMSO or tunicamycin, as expected, we see an increase in the GRIP78, and this is independent of the genotype, so the ER stress is active in both mouse lines. And then if we look at Stard5, what we see at the RNA level, that in the wild type, we are not detecting an increase in the RNA of Stard5 after tunicomycin treatment 4 days following. And in the knockout mice, we don't have any really detection of the RNA. We confirmed this by Western blot analysis, where you can see the Stard5 protein with the control and tunicomycin treatment, and compared to GRIP78, the control and tunicomycin treatment. We see a robust induction of the GRIP78 in both the wild type and the knockout mice. And again, we have no detection of Stard5 protein in our knockout animals. And we can detect a small but significant increase in the level of STAR protein after tunicomycin treatment. So the robust response we see with our RNA levels and cell lines treated with chemical inducers, we're not recapitulating here in the in vivo and the animal model. So when we looked at this, the body weights of the mice were the same, and the response to tunicomycin in terms of loss of body weight were the same. So there's no overt difference there. So we did preliminary studies doing RT-qPCR analysis to confirm that some of our major targets in cholesterol and fatty acid synthesis were changed between the wild type and the knockout mice. Now we used 5 animals or samples each to do RNA-seq analysis. And shown here is the heat map of the differentially expressed genes, the results that we saw. So if we look first here, and these lines indicate a mixture of the wild type and knockout animals, control animals. And what is readily present is that there was no difference in liver gene expression overtly due simply to the knockout of Stard5. But with ER stress response, we do see changes between the wild type and the knockout animals. So this is ER stress is inducing global changes in the liver transcriptome, and we're seeing distinction between the wild type and the knockout mice. So we're asking now whether these differences in the gene expression that we're observing between the knockout tunicomycin treated animals and the wild type tunicomycin treated animals indicates a Stard5 specific response to ER stress. So now this is just providing the summary of differentially expressed genes between the knockout and the wild type. And again, confirming this, and I'll point out here that our cutoff is all those DEGs that are above log 2 fold change of 1. And you can see the significance values, and that had counts of greater than 100 across the samples. So we have I'll say a fairly stringent cutoff that we're using to look at the differentially expressed genes. And then under these conditions, again, as we saw in the heat map, there's really no difference between the knockout and wild type animals. And this one that is down, guess which one that is? Stard5, so we can confirm that. And then you can see here with the numbers, we have over 3,000 in the wild type animals, and this indicates the number of up and down regulated genes, 867 in the knockout animals. And then to get a quick look to see of a genotype dependent difference, we compared the knockout tunicomycin treated counts versus the wild type tunicomycin treated counts to get that differential gene expression. So we can see that there's about 1,097 genes, and you can see the number up and down regulated. So the first step here, we did pathway enrichment analysis to look to see what is the tunicomycin dependent effect on the wild type and the knockout animals. And if you look through this list, you might see some of your favorite pathways. I mean, with the PXR mediated regulation, you can see here it's this transcriptional control of cholesterol and fatty acid biosynthesis, which is what we would expect, ER stress and obesity and type 2 diabetes, immune response, IL-6 induced acute phase response in hepatocytes, these are what would be expected due to the ER stress response. And if you look over at the, this is in the knockout, and if you look at the wild type animals, again, we're seeing those same common pathways that are within the top 10 pathway enrichment analysis. And that's just basically shown down here. We can see what is shared between those two groups is a control of cholesterol and fatty acid synthesis, ER stress, and IL-6 induced responses. So this is then looking at the knockout versus the wild type tuning. Again, here, this is trying to see what is different between those two genotypes. And what we see here, we have, again, we're pulling up the cholesterol fatty acid biosynthesis, IL-6 response, ER stress, and obesity and type 2 diabetes. So those are some of the top pathways that we pulled up, and even though they are tunicomycin, they're the top pathways that are induced by tunicomycin treatment, we're also seeing a genotype-dependent difference between these two. And we're also pulling up some unique pathways. So parsing out what is between the knockout and wild type differentially expressed genes and to those that are upregulated versus downregulated, you can see that control of cholesterol and fatty acid biosynthesis is the top pathway identified that is upregulated. Cholesterol biosynthesis here. And again, the immune response, IL-6 induced acute phase response, is the top pathway that is downregulated in this analysis. So since I am new at looking at all this differential gene expression and trying to analyze these multiple genes, I wanted to confirm this. So I did a compare analysis, which should give me the same information, and it did. So we're looking at compare analysis to try to identify the unique genes in the knockout and wild type and the common genes shared between the two genotypes. And this is indeed what we see is that the unique genes coming out with the knockout animals are involved in cell cycle regulation. The common genes, as we saw, were within the cholesterol, ER stress, and IL-6 induced pathways. And then here are the pathways that were unique to the wild type animals. And the HIF1 targets are also, I think, a common pathway active in NAFLD. So I wanted to then look at the specific genes, what I've provided here in a heat map, to look at how they are specific, to represent how they're specifically expressed. And so what we can see is in the case of the knockout animals, if we look at these are genes within the cholesterol synthesis pathway, tunicomycin treatment had a modest to no effect on genes within this pathway. However, in the wild type mice, we get a significant repression. So when we look at compare our knockout to wild type tunicomycin treated samples, we observe an increase indicating that in the knockout animals, this pathway is more active than in the wild type animals. If we look at the fatty acid synthesis genes, we can see here that we have differential expression between some of the transcriptional regulators, PPAR alpha and SREBPF1, and we have similar expression of the enzymes within the pathway, such as fatty acid synthase and steryl-CoA desaturase. So if we look at this in the where these enzymes lie in cholesterol biosynthesis and fatty acid biosynthesis, you can see all these genes that are within cholesterol biosynthetic pathway from acetyl-CoA down to cholesterol are suppressed in the wild type animals, modestly or not suppressed in the knockout. So when we look at the difference, we see that this pathway overall is up in the knockout animals. Same for the, as I just said, for the transcriptional regulators, whereas if we look at where fatty acid synthase to form palmitate, and then you have desaturation or elongation of those fatty acids, those enzymes are shown here, and you can see they are overall suppressed. So this is indicating that even though we identified these common pathways due to tunicomycin treatment, the knockout mice are responding differently than the wild type mice. And that's what I'm just. So, and how are they responding differently is that the pathways are suppressed in the tunicomycin-treated wild-type mice. So to confirm that we're seeing what we see at the gene level, we can observe at the protein level, we picked some of the targets, Stard4, IDI1, and fatty acid synthase, because there's differential expression between IDI1, similar expression between fatty acid synthase, and did Western blots. And so you can see here with Stard4, we get a marked decrease of the wild-type mice, less so with Stard4. Quantification is over here, so this is not significant. IDI1, again, in the wild-type mice with tunicomycin treatment, that is, expression is depleted, whereas we still retain expression of IDI1 in the knockout mice. But with fatty acid synthase, we see it's lost in both the wild-type and knockout mice. So indicating, and again, here's a quantitation of the protein data, indicating that we have differential expression of these proteins between the two genotypes, and that our protein expression is consistent with the DEG data. So, I'm going to move more quickly and show you that this is looking at hepatic lipid levels. We did oil redo staining, and no question that with tunicomycin treatment, we have an increase in neutral lipids in the liver as expected. We did not quantify the oil redo staining, but we did do direct measurements of cholesterol and triacylglycerols. And what we observed is that with the total cholesterol, we can see that in the knockout mice, we have increase in cholesterol. We see a trending with the wild-type mice, but only significant in the knockout animals. And what we've shown here is the values, and then this is when we express it relative to wild-type DMSO control. So you can see the significant increase in the wild-type mice. And if you look at triacylglycerol levels, this is more evident that you can see that in the knockout mice that we have an increase in tags in the liver. And I also point out we see a lot more heterogeneity in these knockout mouse livers than we do in the others in terms of triacylglycerol content. So hepatic lipid levels in our liver transcriptome support changes in more active lipid synthesis in the knockout mice under ER stress. And then we used the process of liver for CAS staining and H&E staining, and these livers were scored by Dr. Yan Lee. And looking, I can't see up there, but looking at the arrows are indicating the macrovascular steatotic hepatocytes, and the arrowheads are indicating the macrophages. And doing this quantification blinded, what he saw with the wild-type mice and knockout mice with tunicomycin treatment, we do see an increase in the percent of steatotic hepatocytes, but that in the knockout mice, this is more severe. Similarly, with PAS-positive macrophages, we see that they're increased by tunicomycin, as you would expect, but in the knockout mice, there's greater percentage. Using H&E staining to look at cellular damage or lobular inflammation. Again, there's a lot of variability within these treated mice or the knockout mice, and there was no significant difference that was detected. So we are focusing here now that they're consistent with what we're seeing with the cholesterol and triacylglycerol levels, that we're seeing more markings of steatosis in the knockout animals. So membrane, cholesterol, and hepatic inflammation. So what is Stard5 doing? If it's functioning to move cholesterol, this is just shown in a macrophage to the plasma membrane for efflux. It does the presence of Stard5 help to decrease the ER cholesterol load. And then just very briefly, as I introduce this too, because if we have increase in cholesterol in any of these membranes, whether it's the plasma membrane or mitochondria or ER, this is called lipid droplets. These downstream pathways have been reported in NAFLD studies that can induce stellate cell activation, inflammation, Kupffer cell activation and fibrosis or apoptosis. So the question now is this going to be observed in a relevant model of non-alcoholic fatty liver disease? So compared to wild-type mice, what we can say, the Stard5 mice are less responsive to ER stress when it comes to focusing on induced changes in lipid metabolism gene expression, yet they're more susceptible to hepatic lipid accumulation and steatosis. So our proposal is that the Stard5 mice knockout mice have reduced capacity for hepatic recovery after this induction of ER stress. And since ER stress is common to the NAFLD-NASH disease progression, our future goals are to determine or to use these knockout mouse models in a Western diet or Western diet plus PCB-induced NASH development. And due to the time, I'm going to say why PCBs, this is very quick, is that we have modeled that PCBs can bind into the binding pocket of Stard5. So this is molecular docking that was performed by Mark de la Serna when he was here, and that we can look at the binding, I would say affinity, docking affinity. It's not a true affinity, but as you can see, looking at these numbers, these are very, very similar to what was observed or modeled for cholesterol binding into this binding pocket. So now the question is, does Stard5 bind PCBs? And to do that, the second part of the pilot project was to purify Stard5 and to perform PCB binding studies. I won't go through the data due to the time, but we have purified the protein and we are currently doing the binding studies. And I'm waiting anxiously every day to see an email come through to get those data. So, yeah. So what is the model for Stard5 and NASH? Again, here, this is kind of the idea where you have this cyclic pathway dysregulated, where you have a Western diet inducing lipotoxicity that can induce ER stress, and that you have an increase in cholesterol that can promote ER stress. But if Stard5 is present, that cholesterol can be transported to the plasma membrane for efflux to reduce the cholesterol accumulation within the cell. But in the context of this, it's going to be time dependent because cholesterol accumulation within the plasma membrane has been shown to suppress EGFR receptor signaling and activate toll-like receptor signaling. So together, this can promote a transcriptional reprogramming that can promote proliferation, inflammation, and fibrosis. So in the absence of Stard5, if we have less, I would say it's not simply getting cholesterol to the plasma membrane, but probably an interaction of Stard5 with the protein there promoting efflux. So if you don't have Stard5 and you have accumulation of cholesterol within the plasma membrane, you can have potential promoting this disease progression. So the goal here is to use the mouse model, treat it with the Western diet, look at time development of NAFLD and NASH using the standard approaches, and to see if we can delay, see if the absence of Stard5 is going to promote that disease progression, and then we can start looking more at what Stard5 might be doing in the hepatocyte or macrophage or Kupffer cell or stellate cell. I want to acknowledge Dr. Kling, who's been a long-time collaborator and inspiration and has helped move this project along. None of this would be possible without Kelly Peel. She has been a technician with Dr. Kling for many years and worked on the pilot project grant with me, and basically everything I've showed you is the result of her hands. And so excellent collaborator. I want to thank Ron Gregg and Tim Hoffman. Part of the funding from the BMG department was to generate the Stard5 mouse line And the first mouse wrangling was actually done by Dr. Gregg, and now Kelly has taken that over. Again, none of this would be possible without the bioinformatics that we have here currently. And this first, the voucher was for the work of Sabine Weigel, who did the help with the RNA-seq data and analysis by Sheshwa and Jim and Pan. And then Dr. Rushka and De Silva took over in helping with the gene analysis. I identified Mark for the molecular modeling. I do want to highlight two summer medical students who were important for doing some of the first work on ER stress-induced Stard5 differential gene expression in the mice by Vishnu Patel, and then Sydney Travers, who worked on and developed the purification scheme for Stard5 protein. And now again, it's in the hands, the protein expression core help perfect the purification of the protein. That's Will Holmes, under the direction of Mike Sabo, and now Gabriella Snyder is working on the binding studies in collaboration with Nicola Garbett and Rob Monson. And again, I identified Dr. Lee for doing the histology score. And with that, take any questions.
So, do you have any sense of how Stard5 works to get things from one organelle to the other? Is it interacting with other proteins, or is it just the accumulation increases the passive transport?
Yeah, so I guess, do we have any idea of how Stard5 gets cholesterol from one membrane to another? No, we don't. I mean, and that's the challenge. And from my studies, is it just going to that membrane that has the highest cholesterol? Is it being driven to a membrane because cholesterol is going up? And then what is giving it directionality for movement? We don't know. And so I don't know how ER stress is causing that redistribution of that protein. We have some insight. There's recent data coming out on Stard4, it's also the same thing, that what I think it's going to be similar what we saw. They're identifying a phospholipid interacting region on these Start proteins that might give us some indication that it has a preference for one membrane over the other, because the phospholipid content is different between these membranes, just like cholesterol is. So it could be, or it could be the ratio of cholesterol to a certain phospholipid that is going to determine how it moves through the cell.
Have you ever tried to identify other protein interactions with Stard5?
No, but I think Madhvi has been hammering me to try, or suggesting to do that. But that is, I mean, I think that would be a good idea. Yes.
Is there any knowledge or evidence that Stard4 or D5 might be in extracellular vesicles? If they're associating with the plasma membrane, I wonder if they could be in extracellular vesicles as well, if anybody's looked.
Is there any evidence that the Stard4, Stard5 is in extracellular vesicles? I will say no one has looked. Much of the work on Stard4 comes out from a lab that are experts at looking at sterile trafficking intracellularly. And so they're focused on Stard4. So it's really this intracellular distribution of cholesterol and how you can redistribute cholesterol. If you inject a cell with a bolus of cholesterol, it redistributes appropriately. How does that happen? And so that's kind of that's where that focus is. And that's the group that has identified the phospholipid interacting domain for Stard4. So, but no one's looked at extracellular. I don't know if there's a database out there to look at that.
Yeah, sorry. Quick question. So, do you know of sexual dimorphism in Stard5 expression and activity? And I'm asking this because you're talking about the Western diet and PCB model. So usually with those models, with our high-fat diet model, the high-fat diet kind of runs off any PCB effect in terms of hepatic cholesterol levels. But in my recent best-efficacy model, I'm seeing that with females with the high-fat diet model, we have higher hepatic cholesterol levels, and when we expose them to PCB, and it's a mixture of PCB 126 and a pesticide, the hepatic cholesterol levels even went higher, only in the females, even though it didn't really show much signs of steatosis.
So so the short answer is no. The question was about sexual dimorphism for Stard5 in terms of cholesterol, because you're observing that with, Van Rieda's observing with the female mice that the cholesterol accumulation is greater with high-fat diet and with pesticide PCB exposure than the male mice. And right, so currently, planning for our 01 submission for October, the idea is whether to do male and female mice in combination, but that's a lot of mice, but I mean, but if it answers the question, if it's an important question to answer, we have female mice. We do have tissues, but we haven't really followed up on that. We store everything. We've treated both and we store everything, but we haven't followed up on that. We're thinking of using the Western diet, which would be high fat, high cholesterol, high fructose. So trying to push NASH in the male mice, I guess, in that case. And I don't think there's any studies with PCBs in combination with that type of diet. So that's either good or bad. I'm not sure.
I don't think there are with PCB.
Okay.
There are with other chemicals.
Okay. I thought there was a.
So,
Okay. I'm sorry.
so did you know for GWAS associations and polymorphisms that might be human Stard5 gene and interesting phenotypes?
Well, I have been trying to scan all the databases to get as much information as I can. GWAS, I'm not specific for that, but what Stard5 has been popping up in recently is a gene signature for cancer. Breast cancer, colon cancer. So I didn't show here, I'm looking at the inflammatory genes also to see if they're different proliferative genes. That cell cycle that pulled up as unique for the Stard5 knockout mice. So that's a different question, but I mean, it intrigues me that it's in that gene signature for cancer. And you do see in hepatocellular carcinoma, I would say with Kaplan-Meier plots that we pulled out for liver cancer, this kind of loss of Stard5 is worse prognosis. So it is going to flip. Well, actually, it'd be consistent with what we're seeing is that the knockout is more susceptible. Okay, that's mine.
So there are several databases out there for proteomes, extracellular vesicles.
Okay.
And there's one or two databases that are generic that have aggregated proteomic studies that you could query for Stard5. And
Okay.
just, if it's been identified, select, it would reveal what studies are coming from.
Okay, great. Thank you.
Can you share with us your translational vision for this work? Because you're talking about, high cholesterol. You just mentioned cancer. You're talking about PCB exposure. So I'm wondering how you see this, if you extrapolate it, how you see this work translating into what human population, how you hope that it would improve environmental health in general, like public health impact and such.
Great question. Help me think that the significance of the work is the translation of this. If we show a protective role in these types of mechanisms, but maybe we can use it as a marker for those groups of patients, just like in the cancer. If you have a loss of Stard5, you might be at higher risk for, or lower expression of Stard5, a higher risk for HCC. If we see your Stard5 expression level, the caveat, I'm just twisting here, but the caveat here is I don't think Stard5 on its own expression is going to be a marker. We don't see any difference in the knockout alone. I don't think that Stard5 role is going to be revealed unless there is a stressor to the system. So in that case, we can ask about environmental exposures or diet exposures. And if we, in terms of liver disease, right now I'm thinking about liver disease only in this context. Cancer is in the back of, cancer I think would be a good model if anyone is interested in looking at this, because Dr. Klingon and I did a study on lung cancer years ago, and we saw that Stard5 is absent in the lung cancer cell lines, but it's present in normal human bronchial epithelial cells. So there can be some link to apoptosis here. If I'm getting very diffuse here in my response, because what do we want to do translationally? If it plays a protective role, is there a mechanism that we can do to induce it in patients that are progressing from NAFLD to NASH to try to combat disease? Is it a druggable target for induced expression? We can look at its regulation and maybe identify another target upstream that could be more broadly applicable. So I would think if we have, it's not going to stop it. I'm just thinking if we can get that window moved, left shifted a little bit, that will give us time to identify other treatment options.
We're about 10 minutes over.
Okay, stop in. You just, I'll use your email.